introduce. But anyway, I'm Yoram Rudy. For those who don't know me, I'm Yoram Rudy, and I'm here as a member of this faculty. And one of our speakers canceled, could not make the seminar today, so I sort of jumped in. And I thought I'll use the opportunity to, because I presented this electrocardiographic imaging, I think maybe 2 years ago. But my thinking is to show more current, more recent data that was obtained in my laboratory by several Ph.D. students. I must say all of them are exceptionally bright. I'm very lucky to have such wonderful students in the lab. So what I wanted to talk about is non-invasive electrocardiographic imaging of cardiac arrhythmias. And I put arrhythmias in electrophysiology and arrhythmias because last time I think I emphasized non-invasive imaging of normal sinus rhythm, which was really never done before we've done this several years ago. So just as a brief introduction for the work in the lab, the work in my laboratory basically goes in parallel in two different areas that are starting now to merge together, which is really fascinating for us. And they're supported by two separate NIH grants. One of them is a modeling computational biology grant that supports simulations starting now from the molecular level of ion channel proteins and their movement during gating of ion channels through the function of ion channel and the current that flows through it, integrated into the whole cell function to produce both the cardiac action potential and propagation of the action potential in multicellular tissue. So, for example, Dr. Nirbon here is studying the properties of different ion channels, and we can take those properties, and so does Dr. Sui, we can take those properties, put them into the cell, into the cardiac cell, and ask ourselves what will be the consequence of a particular ion channel abnormality. Catherine Yamada, who is here, is studying more of the interaction and communication between cells through gap junctions, and by putting this into this multicellular tissue model, we can also answer the question, how would a mutation or a drug that affects the sodium channel influence conduction of the cardiac impulse in the multicellular tissue. So that's one effort in the lab. In parallel, we've been for many years now developing a novel imaging modality, non-invasive imaging modality, for the electrical activity of the heart. So here we simulate and try to understand mechanisms in models based mostly on animal experiments. And here we try to image and study the mechanisms of these arrhythmias and their presentation in humans in a non-invasive fashion. So I'll focus on this part today. And first of all, we all know that the current method of non-invasive diagnosis of ECGs of the cardiac arrhythmias is the ECG. And the ECG suffers from many limitations. It's been a fantastic tool and has really served the clinical community and cardiologists for many, many, many years now, over 104 years. I think it was 4 years ago, exactly when we celebrated 100 years to the ECG in the Netherlands, where it was invented by Eindhoven. And this is one of Eindhoven patients, and you can see that what you measure is not the heart electrical activity, but the reflection of the heart electrical activity far away from the heart. At that time, it was the extremities. If you have electrodes, in this case saline solution, and you put your hands and legs in it, you will see, of course, the ECG as measured on these extremities. Now we put electrodes on the chest, a little closer to the heart, but still, this is not on the heart surface where you really want to measure. So it has several limitations. First of all, it lacks in sensitivity. It cannot detect arrhythmogenic substrate in many cases. You know, somebody has a heart attack, and that leaves behind a scar, and the scar has arrhythmogenic properties and will generate arrhythmias and sudden death. Most of the time, the ECG cannot detect with sufficient accuracy or sufficient detail. In addition to that, it lacks specificity. It cannot provide specific diagnosis, and in these days of understanding of mechanisms that it's so refined, we know mechanisms of the long QT syndrome, one kind of arrhythmia, based on ion channels function and hereditary mutations. And so we know a lot about mechanism at the molecular and cellular level, but the ECG cannot really detect what is going on in the heart with that kind of specificity. And the most important property of the ECG is not just that it's far away. The most important, and what works against us, that when you measure the potential generated by the EKG, by the heart, at any point on the body surface, anywhere, it does not measure the electrical activity under the electrode. It measures the integrated electrical activity over the entire heart. So therefore, geometrical relationships are lost. And if they're lost, you cannot say from the body surface where this thing occurs in the heart. So you have to go back to the heart in order to make those kind of statements. And so that's what we're set to do. And basically, there are several people here who are bread and butter is imaging, medical imaging, and nobody can imagine anymore, any longer, the practice of modern medicine without imaging, without CT, MRI, ultrasound, and the like. But for cardiac electrophysiology and arrhythmias, which is still a major clinical problem, over 400,000 people die every year suddenly in this country alone from rhythm disorders of the heart. Worldwide, it's estimated to be 7 million. It's possible that with the current excitement about the stock market, it will increase. But at this point, it's estimated at 400,000. Despite all of that, we don't have anything better than the ECG that is truly non-invasive. And that's why, in many cases, we have to put catheters into the heart to make more accurate diagnosis than we can do from the body surface. But that's an invasive procedure. So we were set to develop a non-invasive imaging modality that we call now ECG imaging, ECGI, that basically takes information from the body surface non-invasively and projects it back to the heart. And we've developed it over many years. I think this grant is now its 25th year and still continues. But it takes that kind of a long-term effort to develop an imaging modality that you can trust, because it takes a lot of validation over many, many years. And what you see here is a fantastic collaboration that we had with Bruno Teccardi, who was first at the University of Parma in Italy and then moved to Salt Lake City. Bruno is 86 years old now, and he just retired, went back to Italy, and until last year he was still doing dog experiments, on 2 hands. That's how he is. So what you see here is a fantastic equipment for us to use, where you have a dog heart located inside a torso that is an exact replica of a 10-year-old boy. The reason it's a boy, 10-year-old boy, is because the size ratio between the dog heart and the boy's torso is about the human adult. And what you have are many, many, many rods that carry electrodes on them, so you can simultaneously measure the body surface electrocardiograms all around the torso, and at the same time measure the potential distribution or the electrograms over the heart surface with the tip electrodes that are placed on these rods. And so you have your input non-invasive data, and you have your gold standard, which you want to achieve without penetrating the torso with those rods and without putting electrodes on the heart, but just like CT or MRI, totally non-invasively. And this is just a picture of this thing. The heart is usually put here. It can beat for 5, 6, 7 hours, and you can pace it, and you can put infarcts into it, and you can do a lot of validation using this particular, and that was a key to our success. The first time we applied it in human was in 2004, not too long ago. So it took many years of development and validation in animal experiments before we felt that we are safe enough and secure enough to start doing it in human. And the procedure in human is the following. First, we take 250 electrocardiograms, and you can see here a sample of them. And then, if you sample each electrocardiogram at one instant of time, and you go through all of them, you get the potential distribution at that instant of time over the entire torso for this particular patient. And you do this every millisecond throughout the cardiac cycle. So you have many of those maps, and that constitutes your non-invasive electrophysiological or electrical data set. At the same time, you push the patient through a CT scan that provides you the electrode positions and the heart geometry, also non-invasively. So you have the geometry here, non-invasive, relating the torso surface to the heart surface. You have the body surface potentials, that is the electrical information, and this is where all of our development went into. Developing the mathematics, developing the algorithms, and testing them over and over and over again to make sure that when we marry the two mathematically, we reconstruct the potentials all around the heart and something like 600 electrograms all around the heart. In other words, we reconstruct a very good approximation of what you would have measured if you opened the chest and put a sock with 600 or 800 electrodes on it, but without the need to open the chest. And the last time that I talked about it, which was 2 years ago, I see some people raising eyebrows, like but like G. Miller. But anyway, I'll explain. I'll explain. 2 years ago, I think I went through all of the mathematics of it, at least at some level, and explained that this is an inverse problem, and it requires regularization. It requires a lot of many different mathematical procedure in order to capture what we want to capture. It's a difficult mathematics, and then it's difficult numerical, and then it's difficult algorithms. Everything is difficult. The thing is that this is Mozart when he was 6 years old, and he could do all of this based on his intuition. I mean, this guy was really a genius. I love music, and music, you know, is one of my favorite composers. But he puts all of us to shame. We have to be very humble, because he used to go to the symphony and listen at 6 years old. At least that's a legend. He used to listen to the orchestra and hear the entire integrated music of this orchestra, and then go home, and from memory he would write the score of the different instruments. That's an inverse problem. That's going from the outside, from the hall, to the source of the music, and that's what we're trying to do. We're trying to go from the body surface, which is the whole integrated outcome of the electrical activity of the heart, and try to decipher what is going on in the sections, time, and space in the heart, which basically is the equivalent of these different sections in the orchestra. So he could do an inverse problem intuitively without any mathematical help. We can't do it. We're no geniuses, so we have to resort to mathematical tools. And so validation, I told you, was done in animal experiments, but we also did some validation in humans, comparing to direct mapping during surgery in patients that were undergoing open heart surgery, and they had a sock that we put on, or two plaques. Each one had 100 electrodes. I can show you here. It's the same procedure I just described. You take the body surface potential. At that time we had a vest instead of strips of electrode, but we added 2 patches that we put on the heart directly during open heart surgery in patients. And we measured the electrical activity directly from the heart. And here we reconstructed it non-invasively. So this is the direct measurement, this is the non-invasive reconstruction, and the comparison was our validation. And I just wanted to show you one example. These are electrograms from these locations. The red are invasively directly measured from the heart surface, and the black are non-invasively reconstructed. And you can see that the similarity is really quite good. There are some differences, some of the details are not the same, but for diagnostic purposes, this is really outstanding. So when we saw that, we said, we're ready, and we can start doing it in patients. So to bring all of us to the same page, this is the electrical activity of the normal heart, as measured by Dirk Durer in 1970 in Amsterdam. Durer had 7 patients who died from reasons that were other than heart disease, such as motorcycle accidents, and the heart were donated, and he had lots of needle electrodes and so on, and he measured the electrical activity of these isolated normal human hearts. And what you see is propagation of very broad activation fronts in the left ventricular wall. This is the left ventricle, this is the right ventricle. They go in parallel, that is at the same time. You have activation of the left ventricular wall and activation of the right ventricular wall, propagating from the endocardium, the inner surface, to the epicardium, the outward surface. in very broad, synchronized, and organized activation fronts. And that's key to normal pumping of the heart. An event to remember, for example, is the right ventricular breakthrough. When the activation sequence, when the activation front gets to the right ventricular epicardium, it cannot continue in the human to propagate externally from there because the lung is not an excitable tissue. It will not support conduction. It just transmits fields, but it's not excitatory. So there will be a hole formed in the activation wavefronts that is really observable in all of our reconstructions in normal human beings at around 21 milliseconds from the beginning of the QRS of the cardiac cycle. And here is why those broad wavefronts are established, because the electrical activity of the heart starts from the sinus node, which is the normal natural pacemaker of the heart, shown here in blue. It then propagates radially all direction through the atria, and then it arrives at the AV node, where it has a delay that allows blood to go from the atria to the ventricle before the ventricle contracts to push blood through the circulation. So through the AV node, which is the only connection between the atria and the ventricle, it goes to this very fast, specialized Purkinje conduction system to form those very broad wavefronts that then propagate from endocardium to epicardium in the normal heart. So that's the normal sequence. Abnormal sequences, very broadly, in very broad terms, and in very naive terms, are produced by two abnormal mechanisms. That's a very global definition or global description. One of them is arrhythmogenic foci. That is, a group of cells can become abnormal and become a pacemaker that captures the heart away from the sinus node. And you can have more than one. You can have several of them. So several parts of the heart will now beat in asynchrony. Okay? So that's called focal arrhythmogenic ectopic activity. And these cells could survive. For example, it could be surviving cells in a myocardial infarction scar, somebody who had a recent heart attack. And 2 months later, he suddenly dies because of some abnormal activity of cells. The other possibility is that the cells have normal activity, but conduction of the cardiac impulse is abnormal, and it's very slow, and it's so slow that it can return on its own steps, its own footsteps, and make closed circles known as reentry circuits. And this reentry circuit, you can imagine, emanates wavefronts to the rest of the heart, and it becomes basically an oscillator that drives the heart. And if it breaks into many such oscillators, then you have what's called fibrillation. Every part of the heart will be beating to its own drum. There'll be no synchrony, no pumping, and that's the end. So just remember, there is ectopic activity and there is reentrant activity as mechanisms underlying arrhythmias. So I basically, what I wanted to do is I wanted to show examples of electrocardiographic imaging. And I mostly wanted to focus on recent data, things that I haven't shown in the past. Although it's a mixture, some of them are older, some of them are newer, but I tried to include some new data that we really have not yet published. And this is data published, but I first wanted to show examples of atrial arrhythmias. I'm going from, in the order of the cardiac impulse, from the atria to the ventricle. And so atrial flutter is an abnormality, abnormal rhythm that affects a lot of older people. Atrial tachycardia is not as common as atrial flutter, but still a common rhythm disorder of the heart. And atrial fibrillation is really very common. Atrial fibrillation is becoming the disease, the cardiovascular or cardiology arrhythmia disease of let's say, the next decade. That's clear, because as the population is aging, this is a disease of old age, 70 and older, and there is high percentage of the population suffer from atrial fibrillation. It's debilitating. It's not life-threatening most of the time, but it could cause, for example, thromboembolisms and stroke, but it's a debilitating disease. So here's a case of atrial flutter. First of all, this is an image of normal atrial activity from one of the students who was in the lab when I was still at Case Western Reserve University. And you can see that it starts from the root of the aorta,
That's the earliest in time. This shows isochrones, so these are times of activation. Starts from where it should start, from the aorta, from the sinus node, which is the pacemaker of the heart, and it propagates radially in all directions until the last area to activate in the atrium is the left atrial appendage, shown here in blue, which is late. Okay, so that's normal. This is a patient that presented to us still at Case Western Reserve University with chronic atrial flutter. So his atria was beating at fast rate and abnormally, and that was a permanent situation, a chronic situation for this person. And what you see here is a totally different sequence of activation. What you see here in black arrows is what I showed you before, the reentry circuit or the oscillator that drives this arrhythmia. You see that there is movement of the activation front down the free wall of the right atrium. This is the right atrium. And it then slows down, as you can see from the crowding of this isochrone, as it goes through this narrow isthmus between the IVC and the TA. It then penetrates the septum and comes out very close to what's known as the Bachmann bundle. And that goes on and on and on and on every cycle, and it emanates secondary wavelets. For example, there is one major one that goes around the IVC, comes up, and collides with other wavelets that are emanated from the major rotor or the major reentry pathway. And then it continues to go all the way to the left atrial appendage. So here is a non-invasive image of the electrical activity of this abnormal rhythm. And what was done based on those studies and then validation with catheters was to basically ablate and burn the tissue right here, this narrow isthmus, which broke the main circle of the arrhythmia, the driver of the arrhythmia. If you don't allow it to go through this narrow isthmus, it will stop. And that was stopped, and this patient returned to normal sinus rhythm. As far as I know, it's 6 years now, and I last talked to Al Waldo, I said it's fine. There's no recurrences. Here's a case that we did here together with Bruce Lindsay when he was still here, and most of the work was really done by Yong Wang, who is here. Yong has done a lot of the work that I'll show you, together with Phil Kukulich, who used to be a fellow in cardiology. I think I don't know if he's already faculty, but he will be soon. So this is a case, it's an interesting case. It's a 50-year-old lady who had atrial fibrillation. Not here, she was not treated here, she was treated somewhere else. And she had 2 ablation treatments for her atrial fibrillation. But then a few months later, she developed what's called atrial tachycardia. And it was not clear where it's coming from, so we did this non-invasive image, and what we saw was an intense minimum right here on the roof of the atria between all the pulmonary veins. See, there is a very strong minimum. You can think about why a minimum. If you excite a piece of tissue, there will be an activation front propagating away from it. This activation front is basically electrically a layer of dipoles. So ahead of the front, you see the positive head of those dipoles, and behind them, you see the negative tail of these dipoles. If you sit on the point of initiation, all you see is the negative end of these dipoles going away from you. So you expect to have a local intense minimum at the initiation point. And then that was also verified during repolarization of the atrium, where you see exactly the same pattern, but with reverse polarity, as expected. So that confirmed the location. And here are isochrones, or activation sequences. These are potentials. These are activation sequence. And you see earliest activation, again, at the same location. So all of these are confirmatory as to the location of this thing. This is a scar tissue that was mostly caused by the previous ablations. So ablations can help, but they can also produce substrate that can support arrhythmias. All right? And then Bruce Lindsay went in with a catheter and ablated around this location and at this location, and very consistently, ablation at this location stopped the atrial tachycardia. More complex and more recent data, also from Young and Phil, is the mechanism of atrial fibrillation. We're trying to study the mechanism of atrial fibrillation in patients. It's not really known. You know, you call atrial fibrillation everything that has to do with irregular rhythm of the atria. And from animal models, there are all kinds of mechanisms that they account and be responsible for that kind of behavior of the atria. And there are many, many animal models and many assumptions and many proposed mechanisms. But there's really no good assessment of this in humans, in the human disease, because there was no tool available like ECGI to do that. So we were set to do it. I think by now we have something like 40 patients, you know, something like that. Yeah, we have 40 patients, and we're preparing to write the paper. And the paper is chaotic. All it tells you is that atrial fibrillation is something we suspected is a big umbrella name for many different mechanisms. And in order to treat those patients individually, in what people now call individualized medicine, this is even more urgent. I mean, they have different mechanisms. If you know the mechanisms, you probably can treat them much better than you treat them one size fits all. So we're trying to put some order into this whole story of atrial fibrillation. The mechanisms that were proposed are initiation from the ganglionic plexi, where you have the nervous system coming into the atria. That's mostly supported by the people from Oklahoma City. And then there is what I told you about reentrant activity and the theory of multiple wavelets of reentry causing irregular beating and disorganized beating of the atria. Then there is, of course, the possibility of focal triggers of arrhythmias, and many of them were traced to the pulmonary veins. For some reason, the tissue in the pulmonary veins and the pulmonary vein orifice, where there's some excitable tissue, somehow it's more sensitive to changes, remodeling, aging, and produces triggers more than other. That's not to say that other parts of the atria do not, but the pulmonary veins seem to produce more of those triggers activity. And of course, to make it more complicated, all of those can play. And you can have a very complex situation where all of them are playing together, which is probably the case in many of those situations. So the clinical classification of atrial fibrillation is totally not based on mechanism. It's based on the history of the patient. It's called paroxysmal if it's not permanent, and if it, you know, now you see it, now you don't, kind of a thing, and it doesn't last long, and so on. That's paroxysmal, and it's the easy one to treat, and is the one that with catheter ablation we have good success. Once you get into the long-standing and persistent and permanent, after 4 years, I was in Bordeaux with Michel Hasseger, who is considered to be the master of ablation of atrial fibrillation. They can never succeed in somebody who had persistent atrial fibrillation longer than 5 years. They can slow the rhythm, they bring them back, they do it 6 times, 7 times. I really felt for those patients. It's really torturous. And then the outcome is they don't convert it to normal sinus rhythm. They can slow the rhythm somewhat. Okay? So definitely there is a progression here that nobody really understands very well. So we need to know each individual AF mechanism, AF is atrial fibrillation mechanism, to tailor treatment, drug therapy, ablation, surgical treatment. And ECGI, we hope, could help in this regard. And so I just wanted to show some examples that Young and Field constructed of atrial fibrillation. So this is one that is paroxysmal, and as paroxysmal, it's not long-lasting, it's not permanent, and you expect it to have a simpler, if you will, pattern of excitation. And if I can get, and here's a movie, but before I do the movie, here is the activation sequence that we see in about 70% of the beats. It starts from a trigger activity from the left inferior pulmonary vein. Okay, that's early, red. It propagates radially and then initiates and triggers a very wide, sweeping sort of reentry a rotor that goes around the atria and return. And then another trigger generates the same thing. And that's about 70% of the beats. So you would do a lot of good if you were to isolate this pulmonary vein electrically by ablation, for example. Give you some plan of what you want to do as treatment. And here is a movie of the same thing. And you can see, if you look, you'll see that there will be a circular thing starting from here from a trigger, okay? And then it generates this very broad re-entry circuit or rotor that will, see, this trigger just came out in a very radial fashion from here, and then it generates this very sweeping wavefront goes around the atria. So this was one example of a simple situation. Here's the other extreme. This is an example of persistent atrial fibrillation. And what I show here is an atrial activation movie that Yoram generated. And this is persistent AF for several years, and you'll see that it's totally different from this very organized atrial fibrillation in the paroxysmal case that I just showed you. So mechanisms can be very different depending on the history. And here's this movie. You'll see right away, I mean, this is very complicated. You see everything here. You see a combination of rotors. The rotors break. There are breaking points, there are pivot points, and then there are triggers. They all have players. Very complicated case. And this is because after many, many years, 5 years, 6 years, 7 years of persistent atrial fibrillation, the atria go through a remodeling process. Ion channels change their properties because of remodeling, because they have to adjust somehow to this whole new physiological environment. And the atria themselves, the tissue, becomes more fibrotic, it develops fibrous tissue, and all of that. So now you have an atrium that is very different from the normal in terms of both its electrical properties but also structural properties. And all of those connective tissue that is developed and scar tissue that has developed, all of that breaks waves and provides very complicated substrate for that kind of complicated arrhythmia. So this has not been published yet, I just wanted you to see it. But in any event, so far, we see that in the AF study, we see everything. We see many mechanisms. We see pulmonary vein foci, just like I showed you before. We see non-pulmonary vein foci in the vena cava coronary sinus. Non-anchored rotors that travel through the atria, multiple wavelets, single wave reentry that is sweeping around the atria. And between paroxysmal and permanent, very different properties. One is relatively simple, and the other one, the permanent, is very complex and apparently involves many different mechanisms. So this story is to be continued. I'll go now to the ventricles, and I wanted to show a case of Wolff-Parkinson-White syndrome and cardiac memory, then abnormal electrophysiological substrate after myocardial infarction, a heart attack, and then ventricular tachycardia case. And so again, this is normal activation, very organized, broad wavefronts propagation from endocardium to epicardium. Okay? And the last area to activate is this base of the left ventricle. The base of the left ventricle is the latest to be activated. And here is the right ventricular breakthrough, which is the first thing you see on the epicardium as epicardial activation. And these are comparison of isochrones, activation sequences that we imaged on the heart in comparison to those Dürer isolated humans for normal activity. This is normal. And you can see, for example, here, Dürer has a whole elongated region parallel to the left anterior descending coronary artery where left ventricular breakthrough occurs. That's the beginning of epicardial activation. And then it goes around the heart and from apex to base and activates last the base of the left ventricle. And you can see the correspondence between this, for example, invasive image in the isolated normal heart and our non-invasive reconstruction that shows a similar elongated region along the parallel to the LAD, and then last area to activate at the base of the left ventricle. So that's normal activity. This is Wolff-Parkinson-White syndrome. In Wolff-Parkinson, remember I told you that in normal activity there is only one bridge that connects the atria to the ventricles. And then it generates wavefronts that propagate from endocardium to epicardium. In Wolff-Parkinson-White syndrome, people have a second, another path that allows electrical activity to penetrate the ventricles or connect the ventricles and the atria. And so In many cases, activation will go through here first, and you'll see an area of pre-excitation that will activate fast abnormally in this region before the AV junction with its delay allows normal activation to produce those broad waveforms. This Wolff-Parkinson-White case, this area here that is activated abnormally early is called the area of pre-excitation. The danger with it is that it could be inverted. Activation could go in through the normal pathway, the AV node, come up, and then penetrates the atrium through this unwanted pathway, this accessory pathway, okay? And this accessory pathway, also called the bundle of Kent, provides now a closing of a re-entry circuit. And this re-entry circuit that is now formed can drive the heart in an abnormal rhythm, causing arrhythmia and sudden death. So if you know that somebody has WPW, the treatment today is to simply go in with a catheter and burn it. Okay? You just burn it. So we did many, I don't know, I forget how many. Shubham Ghosh, who is the Ph.D. student in the lab, who did this work together with Jennifer Avari. Jennifer was a fellow in pediatrics, and she's now at Boston Children's Hospital, hoping that she'll come back here. But Shubham is now on vacation in India, and so he did this work. And you can see here an image of one of our Wolff-Parkinson-White syndrome patients. They're mostly pediatric patients. And this shows activation sequences for these before and after ablation. And you see here early activation, totally abnormal. Remember, normal goes from here towards the base. But here, there is a whole region that is excited early, the area of pre-excitation, okay? And once it's ablated, we do the image again, and all of a sudden this area that was the earliest and the source of activity in the ventricle becomes the latest, like it should be. So this immediately reverted to normal rhythm, normal activation that doesn't go from base to apex, but goes from apex to base. Okay? So ablation succeeded, and this patient is cured. Now what's happening to repolarization? There is another example. This is an interesting example. This example shows activation voltages. This is voltages now, not times. And these are voltages before ablation, okay? You see here. And this is repolarization before ablation. So this patient has the WPW case active and pre-excitation region. What was interesting is that they thought, based on the ECG and then characterization, that he had one bypass thread. And we said no. He has one minimum here and another minimum here, and he has two bypass threads. And then when you look at repolarization, that confirms and affirms that kind of prediction, because you have two maxima that correspond to the two minima exactly. So that was then validated in ablation, in catheter mapping, and was ablated as these two pathways had to be ablated in order for this person to come back to normal sinus rhythm. Now what interested us in this whole story is not only what happens to activation, but also what happens to repolarization. And these slides are a little complicated, but it shows activation recovery intervals. If this is the QRS, this is the time of activation. Okay? This is the T wave, is the time of repolarization. So the distance between them in time, this interval, is activation recovery interval. Now, on the electrogram, activation corresponds to the rising phase of an action potential underneath this electrode, and the T wave corresponds to the downslope or repolarization of the action potential. So activation recovery interval is a good surrogate of the action potential duration at that location. Okay? So you see an interesting thing. What you see is that the activation recovery intervals that are plotted here are very heterogeneous, very long at the region of pre-excitation. You see it has the same pattern of these two regions here, and it's very long compared to normal. It is 350 milliseconds, and in normals we've measured 300 milliseconds. So it's 50 millisecond prolongation of the action potential at the region of pre-excitation. Something there is not normal. Okay? Then I told you, you ablate, and activation returns immediately to normal. But look at repolarization right after ablation. it stays the same, basically the same. 45 minutes after ablation, it's still abnormal, still a very large gradient of repolarization and action potential durations. As most of you know, having heterogeneities of action potential duration and dispersion of repolarization could be very arrhythmogenic because it's asymmetrical, can cause unidirectional blocks and reentry. So that's a dangerous situation when you have high gradients of action potential duration. But that persists 45 minutes after. Then we call the patient back, we bring them back a week later, and you still have abnormally long action potential here. In fact, what happens is the following. You look at the T wave, it's inverted, still inverted, becomes flatter but still inverted 1 week, but finally becomes upward like it should be only after 1 month. And in fact, you can see here that the delta, the activation recovery interval differences, the gradients, are 110 milliseconds before ablation, that's huge. 114, which is about the same, 45 minutes after ablation. After 1 week, it's 80, and after 1 month, it's 40. And 40 is what we measure in the image in the normal heart. So it takes about a month to come back to normal for the repolarization process. And this is a long process that has been termed cardiac memory, long-term cardiac memory. And this long-term cardiac memory, because it lasts for a whole month, is very consistent with transcriptional reprogramming and remodeling of ion channels. The ion channels that are exposed to abnormal sequences of activation from this bypass track that they are not exposed to during normal activity change their behavior, change their properties, change their ion channel makeup. And that's what's seen here, and it takes a month to go back to normal. Okay, another example of abnormal substrate is the case of myocardial infarction. And myocardial infarction, as you know, causes a scar, and the scar heals. And this is in a healed scar, a scar that's been there for some time in a patient that underwent myocardial infarction. And what Young did, he basically plotted all the electrograms from everywhere and grouped them together in different categories. You see here, normal, full-blown, high amplitude, single deflection electrograms. These are clearly normal electrograms from normal tissue. So H, for example, here is normal tissue. I here is normal tissue. G here is normal tissue. But then there is a whole group, a subset of electrograms that are very low potential. You can compare, these are the same scale, very low magnitude, and long in duration, and fractionated with multiple deflections. You can see them here on a large scale. There are clearly many deflections. That's known as a fractionated electrogram, and it has been recorded from scar tissue invasively and correlated with discontinuous conduction through the scar. What happens is the scar has islands of surviving myocardium, and you excite one island, and then there is a long delay before it transmits to the next island and to the next island, and those multiple deflections basically capture surviving fibers within this milieu of scar tissue. So that's really a signature of a scar. So Young took those and grouped them together and came up with this red region here that you see that is also graded. For example, A, shown here, is abnormal, but not as abnormal as B and C. So the middle of, the center of the scar is much more severe than the periphery. The periphery has been termed by some people as the infarct border zone. And so now, how do you know what you've done corresponds to anything in reality? The other methods that measures and delineates scars is gadolinium delayed contrast MRI. And there are several people here who know about MRI much more than I do. But anyway, we teamed up with Pam Woodard, and we compared it to gadolinium enhanced contrast MRI in these patients. And Young was able to co-register those images with the CT that we use for our own reconstructions. Okay, you can see, it's hard to see, but you can see here the white shiny part. This is the scar, and you assemble all of those together on the epicardial surface from all core sections, and this is the anatomical scar. and then he co-registered it with our CT images, and now you can make the comparison, and you see that the electrophysiological scar, the abnormal electrophysiological substrate, beautifully coincides with the anatomical scar. In fact, it's most of the time larger, depending on the threshold that we choose for those electrograms, but it's larger most of the time because, you know, this is total scar, but at the periphery of the scar, cells are not completely, or the tissue is not completely normal, electrophysiology. And this is an example of VT, ventricular tachycardia, due to reentry in a patient who has a scar in this location. You see, this is the scar location. This patient has an abnormal heart. I forget, was that an MI or was it a cardiomyopathy? I think it was a cardiomyopathy patient. And this cardiomyopathy patient has, of course, abnormal tissue, abnormal heart from cardiomyopathy. It has right bundle branch block. You can see that the left ventricle activates early. Usually the right ventricle is the one that has the first breakthrough and activates early. Here, the left ventricle activates early and then crosses to the left ventricle late. Blue is very late. So this is normal sinus rhythm in these patients. In this patient, this is normal sinus rhythm. Activation from base to apex of the LV in this orientation, okay? And then across the septum late to the RV. That's normal. But then, spontaneously, he will go into a spontaneous arrhythmia, which is called ventricular tachycardia. And the map shows that it generated by a reentry circuit, a major reentry circuit that you see here, okay, that goes clockwise around the scar. So the scar is where the abnormal tissue anchors this particular reentry circuit that becomes the oscillator that drives the heart fast, abnormally, and emanates wavelets in all directions, okay? And this is the scar as obtained from SPECT images that corresponds to the location of the scar here, and this is where it was ablated successfully. And around, they do ablations in this whole region of the scar, and that terminated the arrhythmia. And I can show you a movie of this thing, if it works. So this is the scar, and what we expect to see a rotating wave around the scar, and nicely they were able to capture a VT beat, another VT beat, interspersed by sinus rhythm beat, and then another VT beat. That's a nice story, nice episode to image, and I hope we can see it. Yeah, you see it rotates around the scar clockwise. Here's the second beat, also around the scar. And by the way, there's diastolic, see? There's all kinds of diastolic activity between beats, which is slow conduction. This continues through the scar. Now you expect to have the normal sinus rhythm beat, and then there'll be another reentry beat. All right? So all of that was obtained non-invasively, and ablation through here terminated the reentry. And this is the first reentry I read about. That was many years ago. It still continues. This one you can't terminate. And here's an example of focal arrhythmia, focal VT, in another patient. This patient, okay, this is induced, not spontaneous. This patient was paced from this region here. Okay, this is the pacing. This is a pace beat. All right, and you see the activation goes around the heart. There is a line of block here. Again, it's an abnormal heart. This is non-ischemic cardiomyopathy patient. So there are lots of scar tissue, abnormal heart, and you have the black is a line of block. So the activation has to go around the line of block, goes around the line of block in both directions, and then here it cannot cross. It'll terminate up here where is the last area to activate. So this is the pace beat that supports this heart. And then, okay, we have an S2 beat, which is a premature beat, two of them. See, this is the normal beats, the blue. The black are premature beats, trying to induce this rhythm by prematurely exciting the heart. And what you see here, it's again starting from the same area because we didn't change the location of the pacing site, but the coupling interval is shorter. It occurs on the background of a heart that is not yet fully recovered from the previous beat. As a result, there is functional extension of the lines of block. You see this line of block is still here, okay? But it continues here, but elongates. Look, look. You know, this is functional because the tissue is not fully recovered. And then it has to go around it and come down here and terminate in this location very, very late, which allows time for the tissue here to recover, to accept another beat. But what happens is that now, if you stop pacing, the tissue here spontaneously generates a VT, beat that continues. And you know, this was the last area to activate, so based on our knowledge of physiology, I would assume this is because of triggered activity. Triggered activity. This is the last area to activate. This is where the action potential just terminated, and all of a sudden VT started from exactly that region where the last action potential was. That to me is a sign of signature of triggered activity, triggered by this premature action potential, that then because of the substrate had to propagate in all kinds of ways and being blocked by all kinds of lines of block because of this abnormal tissue. So ablations through these regions, as you see here, that were given around this region here, where the initiation sites were of this rhythm, terminated the rhythm and was successful, or was not successful. Sorry, I got the story wrong. It was not successful because it's clear from our electrograms that were reconstructed in this region here. It was clear that it was not endocardial in origin. It was from that location, but epicardial in origin. So you cannot. We can say from our reconstruction of electrograms locally whether it starts from the epicardium, mid-myocardium, or from the endocardium. If it starts from the endocardium, okay, then you can ablate it from the endocardium. But if it starts from the epicardium, you cannot approach it with a catheter from the endocardium and ablate it. So that did not succeed.
How do you tell it's from the epicardium?
How do we say? How do we tell? If you look at the electrogram locally, remember what I said about activation away from a site? It has to be negative. It cannot have a positive deflection. So if you see a pure Q wave locally at the initiation site, it has to start from the epicardium. If it starts from the endocardium or the mid-myocardium, you'll have an R wave preceding the Q wave. Okay, I'll show you another example of that I showed last time of a case like this that were eventually treated by, I'll show you in a minute, I don't want to. Here's the case. This is a 29-year-old athlete, actually a soccer player from Europe, who came to us with very, very frequent premature ventricular contractions, known as PVCs, just on routine ECG. What happened to this person? Very interesting story. He wanted to change soccer clubs in Europe, and as a prerequisite, he had to go through routine ECG, physical. He did the routine ECG. They said, you know, your heart. They were surprised he's alive because he had 70% of his beats were abnormal beats, PVC. The danger is that if you leave it alone, and he was not asymptomatic, he didn't even feel it. He was so well conditioned. The problem is that if it does stay that way, you always run the risk that one of them will generate, just like I showed you with the premature paced beat, one of them will fall on a substrate that will be, you know, ready to generate an arrhythmia. Then it's too late. Then it's sudden death. So you don't want to keep it the way it is. So anyway, he came and they put him on a treadmill, and during exercise on a treadmill, he immediately developed ventricular tachycardia, which showed how severe the situation was. And he could still run on the treadmill during tachycardia because he was very well conditioned. So anyway, we didn't need, because ECGI is performed during a single beat, we don't need to invoke the tachycardia. We can take one PVC that has the same morphology as the tachycardia. That's enough for us. And then map activation is the same during the tachycardia. So that's what we've done. And you can see here activation starting from the apex and going very uniformly towards the base. And we showed that the earliest activation site was right here. And Marie, if you see here, it starts directly negative, and it's never positive, no initial positive deflection. So we said it was epicardial. They verified it with catheter mapping, and you see again, very similar map, invasive to the non-invasive. But we said it was epicardial. And in fact, they were not successful trying to ablate it from the endocardium. What was even more interesting is when we look back at the CT, we saw that a piece of the wall was missing right here where this ectopic focus was. This patient has what's called diverticulum, and that means missing piece of the wall, literally. That's because of probably an old viral infection at that location. And you see the wall was paper thin, so you couldn't really ablate too much because you can perforate. In other words, it was epicardial. So the story, what do you do? So as of 5-6 years ago, there is a new method that was actually developed by Sosa in Brazil, in São Paulo, that you make a little puncture here, subxiphoid puncture, and you introduce a catheter without the need for surgery, and you approach the epicardium with the catheter, and you can ablate it from the epicardium. So that was done to this patient with some success. The problem is it's very nice in theory, but the phrenic nerve was in the way, and you can't ablate too much if you don't want to hit the phrenic nerve. End of the story, he went back to Europe, had surgery. This was dissected. He has normal PVCs, no more arrhythmia, and of course he has an implanted defibrillator for protection. From what I heard, he's playing soccer again. So that's the story with this guy. And finally, the last thing I wanted to show is this whole story of resynchronization therapy in heart failure. That's a real hot topic. I mean, two hot topics now in clinical electrophysiology. One of them is this, and the other one is atrial fibrillation. And so we tried to see what's going on here. And the story is the following. You know, heart failure, what happens in those patients that have heart failure, most of them have left bundle branch block. Activation of the left bundle, of the left branch is delayed. The left branch is not normal, and therefore activation of the left ventricle is very delayed. And if it's very delayed, the heart loses synchrony. If it loses synchrony, it doesn't pump blood efficiently, and it's heart failure. And so, what is the remedy? The remedy is to put a catheter with or you put a pacing electrode right here, and you put another pacing electrode through the coronary sinus in the left ventricle. You try to put it where the left ventricle is very delayed. And if you pace it faster, then you bring it forward in time, and you hope you'll resume synchrony. So electrically, you're trying to resume synchrony. Now, we don't know much about the electrophysiology and the electrical activation of the heart in heart failure and during cardiac resynchronization therapy because we didn't have ECGI until now. We know a lot about the mechanical behavior from echo, but we don't know. It's an electrical therapy, not a mechanical therapy. So we were trying to see what's going on electrically. So first of all, this shows an example of, we did it in 8 patients, and this shows the native rhythm in 4 of the 8. And there are many interesting observations to me. One of them is that the right ventricle is completely normal. See, each one of them has right ventricular breakthrough, right ventricular breakthrough, right ventricular breakthrough, right ventricular breakthrough. Beautiful. And then the activation spread is very smooth and very fast, no different from normal. So it turns out it's all a disease of the left ventricle, not of the right. That's number one statement. The other statement is that not all just like not all atrial fibrillation is the same, not all heart failure substrate is the same. For example, here, the last area to activate is up here at 134. In these patients, it's here, very different region of the heart. In this patient, it's an island of late activity here, surrounded by an area of very slow conduction and block. And in this guy, it's down here, very different from this one that is superior. So if you really want to tailor therapy to these patients, you want to put an electrode here in this one, and electrode here in this one, electrode here. We want to try and bring forward in time the latest area to activate.
But is ischemic heart failure patients?
These are heart failure patients.
Ischemic
They are ischemic. We didn't differentiate at that study. We did ischemic and non-ischemic. We're trying now to do non-ischemics only here. So anyway, what happened, and in this patient, for example, if you want to try and put your electrode here, in all likelihood, you're going to fail. Because if you put an electrode here, in all likelihood, you won't capture the heart. See, because it's surrounded by area of very slow conduction and almost full surrounding block. So putting an electrode here is probably not going to be an effective thing to do. So it shows you that, number one, the substrate is very different among heart failure patients. And number two, that in order to know what to do, you need to know the substrate and tailor the therapy to the substrate. That's number one observation. The second observation that I wanted to show is two responders. You can see here the native rhythm of this particular person, and here is after CRT, an electrode pacing from this asterisk, another one from this asterisk, and you see that when you paced from here, it captured the entire left ventricle and did away with this region of very slow conduction, because those lines of blocks blocked everything. But if you're inside this region of block, you can capture that part of the heart and actually resume synchronization. And you can just by looking at the colors see that this is synchronized and this is not. In this case, similarly, an electrode from here, totally not at the area of late activation, but an electrode from here was able to capture this whole area that first before was blocked and activation could not get there, but very slow conduction around the heart. Now it gets directly from the spacing electrode and going in this direction and capture this early enough to synchronize the heart. This is an example of failure. 60% or 70% of the cases are successful. There's about 30% failure, which is big. And so why? Part of it is because you don't know where to put your electrodes. And part of it because you don't know what the substrate is. But in this case, just to show a quick example, here, in this case, this is the native rhythm. There's an electrode here and an electrode here. The electrode here did capture this region that is the latest. But because of very abnormal conduction and substrate and a line of block here, the activation had to go all around, come here, and activate this even later than the late during this native rhythm. So what we did, we actually reduced synchrony. And this is an important portion of the left ventricle for contraction and blood pumping. What we did, we actually reduced synchrony. And this patient did not respond. He was a non-responder. And finally, it shows us also that we really don't need to use two electrodes. This shows the same patient with progressively longer delay between activation of the atrium with an electrode and activation of the left ventricle with an electrode. See, what happens is, if you prolong more and more and more, you allow more and more time. Remember, I told you that the right ventricle is normal, and the conduction system to the right ventricle is normal. And there is right ventricular breakthrough in all of these patients. If you pace from the left ventricle alone, which is what we've done here, we didn't do anything with the right ventricular pacing electrode. We left it untouched. If you pace from the left ventricle alone, you revert the left bundle branch block, bundle block pattern to a right bundle block pattern, right? So now the left ventricle is activated first, and then through slow conduction, the right atrium is activated very late. So now you can say, I can put an electrode here and bring it forward in time. I say there's no need, because you have a right ventricle normal conduction system delayed in such a way that you allow enough time for the right ventricle to be activated normally through its normal conduction system. And that's what we've done. Progressively increase the delay until, look, beautiful synchrony with a single electrode on it. All right? So, that's my story, and these are the people who did all the work. And notably, Yoram did a lot of the work I showed today. Shubham has done all of the work in pediatrics. It was basically a Yoram Wang, Phil Kukulich team, and a Shubham Ghosh, Janavari team. And Lily is our system administrator in charge of our computer, but she also is becoming an expert in ECGI, is on the side. So thank you very much. Itzik. You can ask in Hebrew, it's okay. Everybody learns.
So you mentioned several times that the physicians do ablations of burning of the tissue. So maybe I'm not a physician, how do they know where to do it and how big is the scar and how much to burn? And so does your interface provide some sort of simulation tool to know where to burn?
I mean, the atrial fibrillation, they have a routine. Either ablation with a catheter, or even during surgery, the maze procedure. The maze procedure was actually invented here, developed here. What they do, independent of what's happening to this particular patient, all patients with atrial fibrillation get exactly the same treatment. All pulmonary veins are isolated,
they connect them with line, they do all this. But, you know, if you know the substrate ahead of time and you don't do it blindly, maybe you don't have to isolate all the pulmonary vein. Maybe you can preserve a lot of the atria. Maybe in some patients you have to do more than the routine procedure. That's the idea, is not to operate blindly, but to rely on something that will guide you.
Does your interface provide, I mean, could you simulate what would happen if you burn this area?
We don't simulate, we just see what's the substrate before. We can now see it during the procedure, and we can see it after the procedure, and we can follow up a month after or 2 months or whatever. So we now have a non-invasive method to, just like MRI, CT, and it's intervention, could be interventional.
Right.
Could be interventional. You can, on location, ablate and look right away what happened.
So the AF maps were so complicated. I'm wondering whether or not you had any extra challenges when you were validating the atrial, reverse problem, because you have all sorts of different substrates too in terms of the electrical activity in the pulmonary vein sleeves, and how much do all these other structures contribute?
It's absolutely a great question. And you can't really validate, you can't create this situation to measure directly. There's no, but what we did, we paced from in those patients and patients that had those arrhythmias and so on, we paced from many different places, like the pulmonary veins and different regions of the atrium and so on, and we used that to validate our reconstruction. That worked out very well. And that we're now starting to write the paper, and I think that will be an important part of the methodology of this thing, just like you raised. Because in our mind too, we need to, that's what we've done. So with the same substrate and the same thing, we're able to go back and pinpoint the pacing site, and the activation made sense pacing from that locations.
And then in the one of your ventricular, in the MI, your ventricular map, you had that area of scar, the first one you showed, then there was another area F that was kind of farther away. Was that
That's
continuous
an area,
with the scar?
That's an area up on the base. You have to be careful. We know, we're learning too, but up on the base of the heart, towards the right ventricular outflow tract, the electrograms in normal people, because of fat pad and fat tissue and so on, are very long. So it's not a real scar. Emil.
All
the Israelis, you can do it.
The CT scan gives you kind of geographical anatomy of the, and I guess it's not simultaneous with the electrical. No. So, for example, can you perform ECGI as a part of the protocol of a stress exercise test, and then?
Well, it has to be in the same position, in the supine position. And so what we do is we do the CT first, and then we don't change, with the vest on, of course. So the electrodes, everything is on, we don't change it, we don't touch it. If we want to do a follow-up later on, a month from now, we take all kinds of anatomical markers so we can repeat the position of the electrodes, because we don't want to expose patients twice to CT radiation, although the radiation is low. It's less than 1.8 rods, which is low radiation. But still, so we're trying to repeat the position of the electrodes, and the position of the patient has to be supine in the same position. What you can do is the same thing as done, with exercise MRI. They have those bicycles that you do this while you lie inside the tube. So if you want to exercise somebody in that position, you can. But I don't think you can put it on somebody who is running on a treadmill. It's not the position. You have to be in the same position as the CT was taken.
We do the validation studies with open chest procedure to change the boundary conditions. Did you check that with what you could do with the tank studies?
No, I mean, what we've done, we published several papers on this. That worked to our advantage. Because what happens is that independent of the, I think Martin has a very important question that I didn't mention because we dealt with it. There's a lot of papers on that. But what happens is that, the volume conductor between the heart and the torso has inhomogeneities, like the lungs and skeletal muscle and so on. When you open the chest in a patient, you close the chest, what's the rate relationship? Also, the tank doesn't have lungs. So first of all, in our algorithm for reconstruction, we take into account the possibility we can take into account all those different tissues and the bone. It adds the matrix of relationship between the heart and the torso is growing and growing and growing because we do a boundary element approach. So every boundary adds terms. But what works to our advantage is that the inhomogeneities do not change the potential patterns on the heart, the activation sequence, and the electrogram morphologies. What they do change is the absolute magnitude of the electrograms. And the absolute magnitudes is rarely used diagnostically, so that works to our benefit. So in almost all the applications you've seen here, we basically ignored the torso inhomogeneity.
The patterns.
The patterns also, in the scar, we based it on low amplitude, but it's relative to the normal tissue in the same heart. So.
I have a question. I can see why you would want to find out the exact anatomical location for ablating AF or VF or VT, but if you're not going for that and you just want to know, okay, is there a scar, or is there a functional block, line of block, or just something more general, like what is causing this person's VT? Do you need to have the CT? Because I would imagine that most hearts are approximately the same shape.
They're very different. If you go on, if you see, I've seen right now many hearts during open heart surgery. You'd be amazed how different they are. It's just you can't have one heart fits all. So some of them are elongated like that, some of them are flat like that. They're very different from each other, and I know that that kind of an approach that is not individualized to the particular geometry, it's just not going to work. It's not working.
Okay, so you can't use this in like rural population where they don't have CT in some developing country. I mean, it always has to be paired with a CT.
Or in the future, if people develop a better echo, three-dimensional, high-resolution echo that we can do, it can team up with any imaging modality. MRI is not a choice because MRI, a lot of these patients have implanted devices, you can't put them into the magnetic field. And also MRI takes forever. So CT turned out to be, from all of our experiments and all of our, we looked into this in detail, you can imagine, because you don't rush into exposing pictures like this. At this point, there's no hope. All right, thank you very much.